Welcome to the deep dive. Today we're tackling something really exciting. It's the potential for GLP-1 receptor agonists. You know, the drugs making big waves in diabetes and obesity to maybe, just maybe, make a difference in Alzheimer's disease too.
That's right.
And you're here because you want to get the important stuff quickly and thoroughly. So that's our aim. Break down the key developments, explain them clearly, and try not to get lost in too much jargon.
Exactly. And guiding us is the work of Dr. Lotte Bjerre Knudsen. She's a real pioneer here.
Right, Chief Scientific Advisor at Novo Nordisk.
Yes, she's been there since 1989. She has degrees in biotechnology and scientific medical research. Her whole career really has been in drug discovery, loads of patents in the GLP-1 space.
And she's not just, any researcher. She actually co-invented liraglutide.
Yeah, and she led the biology research for both liraglutide and semaglutide, looking at them across, well, a whole range of things: diabetes, obesity, heart issues, kidney disease, and importantly for us today, Alzheimer's.
Wow. And her work's got a lot of recognition, big awards, like the recent Lasker-DeBakey Clinical Award.
Absolutely. It just underlines how significant her contributions have been.
So our mission for this deep dive,
Mm-hmm.
understand the background, the science, really, that led to this big phase 3 clinical program testing semaglutide for Alzheimer's.
And we're basing this mainly on Dr. Knudsen's plenary lecture from the ADPD 2025 meeting. We should mention, of course, she's a full-time employee at Novo Nordisk, a named inventor, though no royalties, and holds employee stock. Standard disclosure.
Got it. And apparently she shared some really new stuff in that talk. Unpublished research.
Yeah, some fascinating mechanistic data. She actually asked people not to share photos of those specific slides, which, tells you it's pretty fresh information.
Right. Cutting edge.
She also pointed people towards the Lasker Foundation homepage. Lots of info there on the early GLP-1 field. And her own Viewpoint article in JAMA gives a great perspective.
And she stressed it's teamwork, right? This success isn't just one person.
Exactly, a huge collaborative effort. And she made the point that we probably haven't even seen the full potential of GLP-1 yet. Still unfolding.
So what are we covering today based on her talk?
Okay, so we'll look at GLP-1 in diabetes and obesity, the basics, then its effects on inflammation, and then the animal model data specifically relating to Alzheimer's.
But no clinical trial results for Alzheimer's yet.
No, exactly. Those results are expected later this year. This is all about the rationale, the science that got us to those trials.
Okay, let's dive in. Dr. Knudsen stressed that GLP-1 has this really complex, multifaceted way it works. What's so unique about it?
Well, what's really fascinating is its role after we eat. It gets released from these special cells in the gut, L cells, and also from neurons in the hindbrain.
So gut and brain.
Both. And it acts as a hormone traveling in the blood and as a neurotransmitter signaling between nerve cells. It hits one specific receptor, the GLP-1 receptor, which is a GPCR, a G protein-coupled receptor. But unlike, say, the insulin receptor, which is kind of everywhere, the GLP-1 receptor is only in very specific spots, matching its functions.
So quite targeted.
Very. And the really remarkable thing is how all these different actions happen sort of simultaneously after a meal. You get the glucose lowering, the fullness signals to the brain, effects on blood pressure, heart rate, and changes in inflammation levels, all interconnected.
Wow, that's a lot going on from one molecule.
It is. Dr. Knudsen really emphasized that there aren't many biological systems with such broad linked effects. It's powerful, but it also makes it complex to study, because pulling apart which effect causes what can be tricky.
So the takeaway is it's versatile, coordinates lots of things at once, powerful, but complex.
That's a great summary.
Now, you mentioned GLP-1s, plural. There are different types.
Yes, definitely. It's a class of medicines.
Okay.
The first one approved was exenatide way back in 2005.
Okay.
But the ones getting a lot of focus now, and central to Dr. Knudsen's work, are molecules with a fatty acid chain attached like liraglutide and semaglutide.
A fatty acid, what does that do?
Well, think of it like an anchor. It helps the drug stick around longer in the body. And crucially, it helps it bind to albumin, a protein in our blood. That binding actually helps it get better access to the brain.
Ah, so that's key for brain effects.
It seems to be. There are also larger molecules like dulaglutide and even some small molecule versions in late-stage trials now.
And does that structure difference, the fatty acid bit, change how well they work?
It seems to, especially for weight loss. Dr. Knudsen highlighted that liraglutide and semaglutide generally lead to much more significant weight loss. We're talking like up to 300% more compared to some others in the class. Better brain targeting seems to be a big part of that.
Okay, let's talk diabetes. That's where GLP-1 really started, right?
Yeah.
How did it change things?
Yeah, the groundwork was back in the 90s. Scientists figured out GLP-1 was this missing incretin.
Incretin.
It's a hormone from the gut. After you eat, it signals the pancreas to release insulin. So clinical trials followed pretty quickly. Dr. Knudsen mentioned Michael Nock's work specifically. He showed that giving GLP-1 to people with diabetes did two key things: boosted insulin when blood sugar was high, and suppressed glucagon, that's a hormone that raises blood sugar.
And
Importantly, both actions were glucose-dependent. They only happened when needed. This was huge because it meant a much lower risk of hypoglycemia, dangerously low blood sugar, which was always a worry with older treatment.
right, that's a major advantage. But it didn't stop at blood sugar, did it? We started hearing about heart and kidney benefits, too.
Exactly. Liraglutide was actually the first GLP-1 to show clear cardiovascular benefits in a big trial published in 2016.
Which was timely because there were some safety concerns around other diabetes drugs then.
Yes, around 2006-2007, regulators started requiring these cardiovascular safety trials for new diabetes drugs. But Novo Nordisk was already looking into this even before that was mandated. They had some promising biomarker data from back in 2008. They saw liraglutide could lower things like PIAMP and CRP.
Which are markers of
Inflammation. So that hinted early on that there might be benefits beyond just glucose control, possibly related to inflammation.
Interesting. And then semaglutide took that even further, especially linking it to obesity.
Absolutely. The SELECT trial was a game changer. It looked at semaglutide in people who are obese but didn't necessarily have diabetes, and it showed cardiovascular benefits directly linked to the weight loss itself.
That's huge.
It really was. Dr. Knudsen called it a landmark trial. First time a weight loss-focused treatment in obese people showed it could also protect their hearts. It really pushed researchers to think beyond just glucose lowering.
And this anti-inflammatory angle you mentioned, that seems to be a really key thread, especially leading towards Alzheimer's.
Definitely. So let's dig into that. Inflammation. Where did the GLP-1 connection come from?
Yeah, what's the evidence?
A lot of the early clues came from animal models. Mice bred to have high cholesterol or problems processing fats, like LDL receptor knockouts and APOE knockouts. In these mice, researchers found semaglutide could actually reduce inflammatory signals inside the plaques in their arteries. And this happened even at doses that didn't massively affect their weight or blood sugar.
Suggesting a direct effect on inflammation itself.
Exactly, not just a side effect of fixing metabolism.
Yeah.
And it wasn't just targeting one specific inflammatory molecule, like one interleukin. It seemed to dampen inflammation across multiple pathways, a broader effect.
So a system-wide dampening. What about in people? Can we see that?
It's trickier to measure plaque inflammation directly in humans, obviously. But Dr. Knudsen shared some data from a study. It wasn't a randomized trial, important caveat, but it compared people taking GLP-1 drugs versus a control group.
Okay.
They looked at markers of plaque stability. The treated group showed a significant increase in the fibrous cap of the plaques.
Which means?
A thicker cap means the plaque is more stable, less likely to rupture and cause a heart attack or stroke. So while not definitive proof, this human data lines up nicely with the animal findings, pointing towards this anti-inflammatory role in heart disease.
Okay, fascinating. Now let's shift gears to the brain. This idea that GLP-1 affects the brain, especially for obesity, that's not new, is it?
Not at all. That link, GLP-1, brain, obesity, that's been researched for, wow, about 30 years now.
Really?
Yeah, even before the diabetes link was fully nailed down, studies back in the 80s found GLP-1 receptors in mouse and rat brains. Then came a really pivotal study in 1996 by Stephen Bloom. He showed if you put GLP-1 directly into the brains of rats, even rats that were starving, they just stopped eating.
Wow. Just shut off the hunger signal.
Pretty much.
Yeah.
And his study showed the GLP-1 was binding to neurons, activating them. Clear evidence of a brain effect on appetite.
And that research directly inspired developing GLP-1s for obesity.
It played a big part. Dr. Knudsen mentioned her own work on liraglutide for weight management was partly inspired by some odd animals with tumors that produced GLP-1. These animals basically wasted away.
A bit grim, but informative.
Right. Now, with semaglutide being so widely used for obesity, we understand a bit more about how it works in people. It clearly reduces energy intake. People feel fuller, less hungry.
And it affects cravings too, right? That's something you hear a lot.
Yes, it seems to impact the brain's reward system. People report better control over cravings. Interestingly, Longer-term data suggests the effect might be stronger and more sustained for savory food cravings compared to sweet ones, but overall, craving reduction is a consistent finding in humans.
Even if some animal models show slightly different things.
Yeah, that's a good point. Sometimes animal models don't perfectly predict human responses, especially with complex things like cravings. The human data shows a consistent, numerical reduction, even if statistical significance varies a bit depending on the study duration and specific craving type.
So how does it actually get into the brain to do this? I thought there was a barrier.
There is the blood-brain barrier, and semaglutide doesn't cross that main barrier easily. But the brain has these special regions called circumventricular organs, or CVOs. Yeah, they're like little windows in the barrier, areas where the brain samples the blood more directly. Semaglutide can access these CVOs.
Ah, yeah.
And from there, it triggers signals. In mice, we see neuronal activation starting in places like the lateral parabrachial nucleus. And then that signal travels deeper into the brain areas controlling energy balance.
So it doesn't need to flood the whole brain, just hit these key access points.
That seems to be the current thinking based on, half a dozen key papers Dr. Knudsen mentioned. GLP-1 agonists access the CVOs and signal from there. There are also neurons originating in the hindbrain that produce GLP-1 and project widely, activating receptors indirectly. So it's this orchestration of signals.
But you mentioned earlier, a lot of this detailed brain mechanism stuff in obesity comes from animal models.
Right. That's a limitation we always have to keep in mind. Animal brains aren't human brains.
But for Alzheimer's research, there's actually more human brain tissue available for study, which must be helpful.
A huge advantage. Dr. Knudsen highlighted a recent Nature paper she co-authored using spatial transcriptomics.
Which lets you see
Which genes are active where in actual human brain tissue. It gives incredible detail for understanding disease mechanisms.
Cool.
And they've also looked at proteomics, the proteins in obese people treated with GLP-1, comparing them to big population data sets like DECODE in Iceland.
And what did that show?
They saw signatures related to other brain conditions, sleep apnea, insomnia, addiction, mood disorders. Interestingly, not a clear Alzheimer's signature, but that might be because the obesity cohort was relatively young.
Right. Age is a big factor for Alzheimer's. And you mentioned some real-world data popping up, too. Hints of benefits beyond diabetes and obesity.
Yeah, it's preliminary but intriguing. Some database studies suggest people on GLP-1s might have lower rates of things like alcohol use disorder hospitalizations or opioid overdoses.
Wow.
Again, these are not randomized controlled trials. They just show associations, and the real effect size might be smaller. But it definitely flags these areas as needing proper investigation. It adds to this picture of broad effects.
Okay, so all of this background, the inflammation angle, the brain effects in obesity, sets the stage for looking at Alzheimer's. When did that specific research direction really start?
It actually goes back further than you might think. Dr. Knudsen mentioned the idea of GLP-1 in neurodegenerative disease surfaced around 2002.
Really? What was the thinking then?
The initial hypothesis was kind of an analogy. GLP-1 seemed to protect the insulin-producing cells in the pancreas. So the thought was maybe it could protect brain cells too, since we knew the receptors were there.
Makes sense.
Then a 2003 paper in Nature Medicine specifically looked at the GLP-1 receptor in the hippocampus key for memory and its potential role in cognition.
And Dr. Knudsen herself started studies around 2010.
Yes, but she was quite candid, saying the early animal model results were a bit messy, inconsistent sometimes, and it wasn't always clear if the cognitive tests in mice really mirrored human Alzheimer's.
So what was the turning point?
She said it was connecting the dots, taking their understanding of GLP-1's anti-inflammatory effects in cardiovascular disease and combining it with this growing evidence about neuroinflammation being central to Alzheimer's. A key 2018 Nature Medicine paper really highlighted neuroinflammation, plus more and more genetic studies were implicating inflammatory genes in Alzheimer's risk.
Ah, so the inflammation link became the bridge.
Exactly. That convergence of evidence really solidified the rationale, and that ultimately led them to launch the big semaglutide trials for Alzheimer's, EVOKE and EVOKE Plus back in 2021.
Okay, so we've seen the journey. Now let's get specific about Alzheimer's. How might GLP-1 actually be working there? What are the proposed mechanisms?
Well, scientists use different frameworks. One is the ATN framework, amyloid, tau, neurodegeneration.
The main pathological hallmarks.
Right. GLP-1 probably doesn't directly attack the amyloid plaques or tau tangles themselves, but the idea is it might reduce the neurodegeneration, the loss of brain cells. It could also potentially help with inflammation and maybe vascular issues in the brain, which could then indirectly affect amyloid and tau pathways downstream.
So indirect effects on A and T, but maybe a more direct effect on neurodegeneration and inflammation.
That's one way to look at it. Another framework is CADT. Cognition, activities of daily living, overall clinical status. Here, GLP-1 seems to touch on many relevant pathways. Dr. Knudsen used the phrase numerous check marks, suggesting broad potential relevance.
But the main hypothesis driving the current trials is neuroinflammation.
That's definitely the lead hypothesis. The idea is that semaglutide's core benefit in Alzheimer's might come from its ability to dampen that harmful inflammation within the brain environment.
And neuroinflammation involves what exactly?
It's complex. It includes things like a leaky blood-brain barrier, oxidative stress, inflammation spilling over from the body, and the activation of the brain's own immune cells, microglia, and other support cells called glial cells. And there are literally hundreds of papers showing GLP-1 can influence these various processes in different models. So the central idea being tested is, by calming this neuroinflammation, semaglutide will slow down the neurodegeneration process that drives Alzheimer's symptoms.
And where are the GLP-1 receptors actually located in the brain to mediate this? On which cells?
Good question. The initial thought was mostly neurons.
The signaling cells.
Right. But more recent, detailed work, like a vascular brain atlas published in Nature in 2022, showed expression elsewhere too. On smooth muscle cells and blood vessels, on ependymal cells lining the brain's fluid spaces, on astrocytes' key support cells, and even on some oligodendrocyte precursors, which make myelin.
Interesting. So not just neurons. What about microglia, the immune cells?
There's literature suggesting receptors are on microglia, but Dr. Knudsen noted some caution there. The techniques used, like immunohistochemistry, can sometimes give false positives, so confirming microglia expression definitively is still a bit tricky.
So the picture of where it acts is still evolving, but it's clearly broader than just neurons.
Exactly. And they're leveraging human brain data heavily now.
How so?
Well, studies using bulk transcriptomics looking at all the RNA messages from big brain bank cohorts like RoseMap and Mount Sinai have actually identified different subtypes of Alzheimer's.
Subtypes?
Yeah. And neuroinflammation seems particularly prominent in the subtypes where amyloid buildup is the main feature. This human data helps inform which animal models might be most relevant.
Which led them to choose a specific model.
Yes. Novo Nordisk made a strategic choice to use a relatively simple model, inducing brain inflammation with LPS, lipopolysaccharide.
LPS, that's like a bacterial toxin.
Exactly. It triggers a strong inflammatory response. The idea wasn't to perfectly mimic all aspects of Alzheimer's with amyloid and tau, but to specifically hone in on the neuroinflammation component and see if semaglutide could counter that.
So, using a focused tool to ask a focused question about mechanism.
Precisely. This LPS model creates neuroinflammation that shares features with what's seen in those human Alzheimer's subtypes, allowing them to study the drug's effect on that specific process.
Okay, so what did they find when they treated these LPS-inflamed mice with semaglutide?
Some really interesting things. First, looking at the microglia, semaglutide treatment helped to normalize their physical area or size, which gets altered during inflammation.
Okay.
And using transcriptomics analyzing the genes, it reduced the number of inflammatory genes that were being switched on together. It sort of quieted that coordinated inflammatory response.
So calming the microglia response.
Yes, and promoting what they called microglia homeostasis, getting them back towards a more balanced, healthy state. They identified specific gene programs related to this, and one key program related to homeostasis was significantly boosted by semaglutide.
Anything else?
Yes, another key finding was about immune cells coming from outside the brain. They saw that semaglutide significantly blocked neutrophils, a type of white blood cell, from infiltrating the brain tissue in these LPS mice.
So reducing inflammation coming from the periphery too.
It seems so, at least for neutrophils, at most time points they looked at after the initial LPS challenge.
You mentioned time points.
Yeah, Dr. Knudsen explained they used relatively short studies, like 2 hours, 5 days, 11 days post-LPS. Her rationale was that GLP-1 mechanisms often act quite quickly, and the goal here was understanding these early mechanistic effects, not long-term cognitive changes in the mice.
Makes sense for mechanism studies. Did they look at the APOE gene interaction at all? That's such a big risk factor.
They did. They looked at the top 50 known Alzheimer's risk genes and checked their activity in different brain cell types, microglia, endothelial cells lining blood vessels, and parasites supporting them. What popped out was that in the microglia, several genes that were significantly downregulated or turned down by semaglutide were located right within the APOE gene region.
Wow. Which gene specifically?
Genes called CLPTM1, RELB, and BCL3. They all sit in that APOE locus chromosome 19 in us, chromosome 7 in mice.
That's fascinating. Is there a connection to what else GLP-1 does?
Potentially, yes. We already know GLP-1 drugs affect lipid metabolism, how the body handles fats. They influence things like ApoB48, ApoB100, triglycerides, all of which are biochemically linked to APOE's function in handling lipids.
So a potential link between GLP-1's metabolic effects and a major Alzheimer's genetic risk pathway.
Exactly. It's a really suggestive finding from the animal work.
Okay, so pulling that animal data together,
Mhm.
what's the working hypothesis now for how semaglutide might be helping in Alzheimer's based on these models?
Based on this animal data, the hypothesis involves several things: alleviating inflammation, particularly in the hippocampus, promoting microglia homeostasis, that healthier state, limiting neutrophil infiltration from the periphery, and interacting with the APOE locus.
But crucially, this is still animal data.
Absolutely crucial point. These are hypotheses generated from animal models. That's why the human trials are so important.
And those trials are looking for these same signals in people.
Yes, they have a very strong biomarker program built in. They'll be looking at omics data, classical Alzheimer's biomarkers in spinal fluid and blood, imaging, and broad, unbiased protein measurements to see if these mechanisms observed in mice are actually playing out in humans receiving the drug.
It sounds incredibly thorough. Finally, Dr. Tinson wrapped up with some broader thoughts on drug discovery from her, 36 years of experience.
Yeah, some really valuable perspective. Her key message was about the importance of deep understanding. To increase your chances of success, you really need to understand the biology at the molecular level, the cellular level, and the whole organism or physiological level.
Connecting the dots across scales.
Precisely. She said GLP-1 was somewhat fortunate because there was already a strong physiological understanding to build upon when developing the pharmacology.
But for brand new targets.
For new targets, you really need to map out the pathway, figure out if it's druggable, and ideally understand its relevance in actual human disease. Using human genetics, human cell data, maybe insights from other clinical data sets, that's key.
And she reiterated that GLP-1 isn't just doing one thing.
Right. It's multifaceted. Several mechanisms likely contribute to the overall outcomes. So while neuroinflammation is the leading hypothesis for Alzheimer's right now, they're not excluding potential roles for metabolic pathways too.
It's about having a primary hypothesis, but keeping an open mind.
Exactly. Having that predefined hypothesis around neuroinflammation is important for testing, but the known cardiovascular benefits, which also involve inflammation, provide strong supporting evidence for relevance in neurodegenerative disease. And she made sure to credit her many colleagues, specifically mentioning Peter Johansson, a leading neurologist involved in the work.
Well, this has been incredibly insightful, really digging into the science behind why these well-known drugs are now being tested for something as complex as Alzheimer's. The neuroinflammation story is compelling.
It really is, and it highlights, as you said, how interconnected things are. Effects seen in diabetes and obesity, particularly around inflammation and metabolism, might have profound implications for the brain.
And as we've stressed, the animal data is promising. It shines a light on potential mechanisms. But the real answers will come from those ongoing Phase 3 trials in people living with Alzheimer's.
Yeah.
Everyone's eagerly awaiting those results.
Absolutely. It's a reminder of the careful, step-by-step process of science. Integrating knowledge, testing rigorously in humans, that's how progress happens.
So the final thought for you listening, given just how broad the impact of GLP-1 seems to be hitting multiple organ systems, multiple diseases, what other unexpected applications might still be out there, it makes you wonder about the hidden connections in our biology and where current therapies might find surprising new roles.